We will close this lecture on glial cell functions by talking about one additional function of glial cells, and specifically astrocytes, and that is the glial cell regulation of blood flow. Now, it's been known for over a century that when a region of the brain is activated, there is a localized increase in blood flow to that region of the brain, bringing added oxygen and glucose to the active neurons. This response, termed functional hyperemia, is the basis of functional brain imaging using such techniques as bold fMRI. So, if one looks at a functional brain image, such as the one shown in this figure, and you see a region of the brain that lights up, you're not looking at increased neuronal activity directly. What you're looking at is increases in blood flow that's caused by the increase in neuronal activity. So there are mechanisms that couple increased neuronal activity to blood flow. Active neurons have to signal to blood vessels, causing them to dilate and thus to bring added blood to that region of the brain. This process is called neurovascular coupling, and it is now believed that glial cells play an important role in mediating neurovascular coupling in the CNS. This occurs by signaling from active neurons to glia, and then glia to blood vessels. So initially, active neurons release neurotransmitters, which stimulate the glial cell, causing a calcium increase in the glial cell. The calcium increase in turn leads to the production and release of vasodilating agents, and these are released onto blood vessels, causing them to dilate and blood flow to increase. Remember that astrocyte processes, the end feet of astrocytes, completely surround blood vessels, so the astrocytes are perfectly situated to signal to the blood vessels. And experiments indicate that one of the principal vasodilating agents that are released from astrocytes are metabolites of arachidonic acid. A similar experiment was done in vivo in the cortex of a rat. In this case, as in the previous example, glial cells in the cortex were labeled both with a calcium indicator dye and with cage calcium. This is shown here. This is a arterial, penetrating arterial that is diving down into the brain. It is surrounded by the end feet of glial cells here, and we can see that they are labeled with the calcium indicator dye. This bottom sets of panels show what happens when the glial cell is stimulated. Here is the blood vessel, again in green, the astrocyte end feet in this golden yellow, and here an end foot is stimulated with a UV uncaging flash, causing a calcium increase in the glial cell processes, and we see that the blood vessel does in fact get larger. It is dilating. This record on the bottom left shows both the calcium increase in the astrocyte and the increase in diameter. Shown in red is the calcium increase. This is the uncaging flash causing a calcium increase in the glial cell, and this is followed very soon after by an increase in the diameter of the arterial, showing that in vivo, as in the retina, as we saw in the previous slide, if one directly stimulates glial cells and causes a calcium increase, this leads to the increase in diameter of arterioles, in this case in the brain in vivo. I mentioned previously that the vasodilating agents released from astrocytes onto blood vessels are thought to be metabolites of arachidonic acid. This figure here shows how production of these metabolites could come about. With neuronal activity, there is a release of neurotransmitters from presynaptic terminals. The main transmitter would be glutamate. This glutamate would activate metabotropic glutamate receptors on the astrocyte. This in turn would lead to an increase in calcium in the astrocyte. This calcium increase would activate the calcium sensitive phospholipase A2, which converts membrane phospholipids to arachidonic acid. And with the increase in arachidonic acid in the astrocyte, metabolites of arachidonic acid, including EATs and prostaglandins, would be produced. These are membrane permeable substances which can diffuse out of the astrocyte through the astrocyte membrane and onto the vascular smooth muscle cells of the arterioles, causing the vessels to dilate. Now, this diagram indicates other possible neurovascular coupling mechanisms that may also be operating in the CNS. With an increase in calcium, this could activate calcium-activated potassium channels in astrocytes, leading to the release of potassium. And potassium, when it is released onto blood vessels, also can cause a dilation of the blood vessel. This diagram also indicates that neurons can also signal directly to blood vessels. They don't have to signal through astrocytes, but may also directly control vessel diameter by a number of mechanisms. One of these is the production of nitric oxide with the activation of metabotropic or in this case NMDA receptors on a neuron. Calcium will increase in the neuron. This will activate nNOS, neuronal nitric oxide synthase, leading to the production of nitric oxide, which can diffuse out of the cell and onto the vascular smooth muscle cell, causing vessels to dilate. And as is shown in the diagram, neurons can also synthesize prostaglandins, which can be released onto vessels and cause vessel dilation. So this diagram illustrates a very important point about neurovascular coupling, that multiple mechanisms undoubtedly mediate neurovascular coupling, but it does emphasize an important point that astrocytes are believed to play an important role in this process of the regulation of blood flow. This brings us to the end of our discussion of the functions of glial cells, and I'd like to close by just summarizing some of the interactions that occur between neurons and glial cells, primarily astrocytes, in the central nervous system. We see here a tripartite synapse with the pre- and post-synaptic components of the neurons, as well as the astrocytic processes that surround the synapse. When a presynaptic neuron is activated and releases glutamate, the removal of glutamate from the synaptic cleft is controlled largely by glutamate transporters on astrocytes, which remove glutamate and take it up, transport it into the astrocyte. Now, the glutamate that's released can activate receptors on the postsynaptic neuron, but as we've seen, this glutamate and other transmitters, including ATP, can also activate receptors on the astrocyte. Activation of these receptors, which are primarily metabotropic receptors, can lead to the production of IP3, which in turn results in the release of calcium from the endoplasmic reticulum, leading to calcium increases in the cytoplasm of the glial cell. These calcium increases in turn lead to the release of gliotransmitters from the glial cell, and these include ATP and glutamate, as well as D-serine. These gliotransmitters can act on receptors on the presynaptic terminals at synapses and in turn control the strength of synaptic transmission and also the strength of synaptic plasticity that is seen at synapses. These gliotransmitters can also act on receptors on the postsynaptic neuron, leading to increases or decreases in the excitability of the neurons. Calcium increases in the glial cells can also be propagated to other glial cells as calcium waves, and this occurs by the diffusion of IP3 through gap junctions to coupled glial cells, and also by the release of the gliotransmitter ATP, which can activate purinergic receptors on secondary glial cells, leading to release of calcium. And finally, calcium increases can lead to the production and release of vasoactive substances such as arachidonic acid metabolites, which can result in the dilation of blood vessels and to local increases in blood flow.